his trajectory. Currently unemployed or searching for employment?
I think the best term is fun employed.
There you go. That's the best way to do it. So if you guys would divert your full attention to him, open later, kind of open forum the last couple of minutes, give him some time for some questions. So,
if you don't mind, actually, just speaking of my question, given how it works out the best, I don't have a prepared spiel at all. I've always been interested in complex systems, grew up in a family of engineers, and then went into biophysical sciences because I had seen Jurassic Park. Like I had to negotiate giving up my eye teeth at the age of 9 to see Jurassic Park in theaters, and like that little DNA dude saying that you can make dinosaurs and stuff. And I was like, all right, let's go see what the science is actually doing. And got into Microbiota at University of Michigan, and it took a while to get into a research laboratory. But once I did, I got into a serious pathogen laboratory, and that introduced me to the field of immunology, which is how your body figures out harmful non-self versus harmful self, and kind of keeps you healthy overall. I mean, pathogens are just part of it. The immune system also helps you heal from injuries. It helps you get rid of cancer, and it forms long-term memory to prevent happening again. But it's probably one of the most messy and complex biological systems that you can apply so many different things to. So it's kept my interest for a long time, and with that, I've done several research positions. Then I got into academia for a while, and joined a robotics startup. And when that crashed, picked up its clients, did it independently, doing kind of similar projects to what you're doing, but more building prototypes for industry for a while. At the same time, built a nonprofit hackerspace to kind of give tools to everyone more freely. But then that got going, and I had realized that I wasn't happy because I was far away from biotech and far away from microbiology. So I was fortunate to meet my co-founder at Xander, who had just built a prototype of this kind of bio-nanotechnology, where he had self-assembling polymers based on their charged properties that form these multi-shelled nanoparticles about 100 nanometers in diameter. And we were able to package genetic material on the inside of these that once it got inside of the cell, would be read by the cell and then do useful things. And like with that, we had injected into mouse femurs and gotten them to produce more bone in those brittle bone disease mice. And now that the company is working with Parker Foundation to take that, going after like HIV vaccine, going after sickle cell anemia, going after immuno-oncology as well. So taking that kind of platform biotechnology, we have to figure out how do you take this new technology that we don't even know how to make at more than 10 micrograms at a time, and produce it to an actual medicine that does things really, really well. And that was an interesting process. Biotechnology allows you to do so much in terms of taking the scientific thread and getting just a bit of proof on the other side of it, and then figuring out a very clear path to bring that proof into reality. It's also one of the most regulated industries on earth, with the FDA and everything you have to do for human trials, but the FDA is made up of scientists, and they're pretty reasonable in what they actually ask you to do. But even so, like a drug at this point costs about $2 to $3 billion to bring to market each, and takes usually about 7 years, and involves easily 3,000 or 4,000 people working on it. There are, however, with the maturation of new tools based on the kind of technology we're working on here, that path is getting more diverse, and it's about to open up in a really big way. So traditionally, only pharma can play in drug development because they're the only ones that have enough money to do it. But now you see other companies like Pearlstein Laboratories in Oakland or Emerald Therapeutics up in San Mateo who are working on making drugs for much, much less money. And they're going after that by choosing very carefully what disease are they treating, designing very carefully their experiments to actually get there, and then utilizing advanced technology that allows them to get more data more cheaply and in less time. So I think there's a lot of things that can happen there, and now that Xander's gotten to a point where it can survive without me, I'm out looking at what I want to do next. And you've seen Robin before. I'm working with Robin now on a possible project that's designed to take in academic research that's not being commercialized and combine it into new products to be kind of a lean incubator model for making biotechnology products faster and better than previously. I'm also looking at other possible biotechnology assets that I could develop, and doing some scouting for biotechnology firms right now who are looking for assets for their pipelines. But I mean, the asset evaluation process itself is always really interesting. Like, if you're doing engineering for robotics, you have to start with your requirements of like, what do you want your robot to do in the end. For a biotechnology product, that has to be what does it do in a patient? What are the acceptable tolerances? What are the acceptable side effects? What's the current standard of care? what will it take for doctors to actually prescribe this new thing over what they already have? So there's a lot of opportunities, I think, especially what we're going to see in cancer, that we're going to see in genetically-defined diseas, and that we're going to see in regenerative medicine coming out soon. Like we actually now see diseases starting to be cured. Like Gilead recently introduced a drug that basically cures hepatitis C, and that is a new thing in biotech. Like actual cures are about to become possible. So I think it's a really exciting time to be working in it. I'm excited, like working with Robin, about what kind of stuff we can do in terms of making more products get to market faster than pharma does on its own. At that point, I don't really have a prepared spiel whatsoever. So any questions you have are completely welcome.
Can you provide students a scale for, so you're talking about 100 micrometers?
100
nanometers.
100 nanometers. So
Can you
that's the
provide
size of a
students kind of-
virus.
Perfect. How about like comparing that to something they would know?
Okay. So you know, so we're down in the completely non-visible scale. So a cell tends to be 10 micrometers long to 100 micrometers long. You have roughly 2 or 3 meters of DNA packed into a very small space. But even like that compaction of that DNA into a small space, that's still on the order of at least 1,000 nanograms for like a microgram for a nucleus size. So we're talking a tenth the size of that for these virus-like particles. And each of those shells can be about 10 nanometers thick. And these are made by taking biopolymers, so amino acids that are in chains that have positive and negative charges where based on how many electrons or neutrons or protons they have balanced where, these things then kind of come together into sticky balls, for lack of a better term. But then by altering the conditions around those sticky balls of polymers, you can then compact them further and make them more robust for getting into the bloodstream and not being destabilized by the enzymes we have there.
Sorry, I didn't really understand. So could you tell me more about your startup with Robin? So yeah, just elaborate on that. Sorry.
So to go into a really high level in terms of how R&D is done in pharma, right now pharma does not do a really good job internally of doing research and development. And that may be inherent to large organizations having slow communication times and having a hard time making decisions. And also incentives get really weird in big organizations because like a lab manager for Roche, they have an incentive for their budget not to be cut. And even if they're not making a lot of science happen, they still need to spend that money, so they wind up buying more robots than they need, or hiring people they don't need, and that just slows the entire process down further and further. So for a long time, pharma's actually looked to biotech startups for bringing assets into their pipeline. So you have most biotech startups probably exit by being acquired by a Roche or Bristol Myers Squibbs, and it's a matter of getting enough proof around your candidates that they want to actually go for it. So they look at it in terms of risk and eventual value down the line. And if you can de-risk your candidate sufficiently, pharma will then be interested in paying a premium for it because it's cheaper for them to spend $20 billion on acquiring a startup than it is for them to try to do it themselves, especially for a very large indication like cardiovascular disease or neurodegeneration. So what we're doing at Robin is we're taking a look at that kind of early stage innovation because there's a big gap going on between academia and startups. So you have some people in academia who are industry-minded and want to do startups, and they do pretty well on their own. But you also have a lot of other academic professors who are not interested in going anywhere near industry. They want to stay professors, they want to do research, and that's great. But they're also producing technology that could be translated into something valuable, and they have no idea how to do it. So we're trying to create a group that has internal competencies on evaluating that technology, and then actually running the experiments that prove it works well enough to either be acquired or be its own company.
Come on, guys. Can't we just be instructors who find this stuff really interesting?
What's a pharma and a Roche?
Sure, sorry about that. So in the industry, you have a division between pharmaceutical companies and biotechnology companies. And you have about 7 large pharmaceutical companies that make the majority of the drugs in the world. And Roche is one of those. It's one of the oldest based out of Switzerland. So like Roche, Pfizer, Eli Lilly, Johnson & Johnson are some of the biggest pharmaceutical firms in the world. And they tend to employ like 250,000 people and spend insane amounts of money on R&D per year, even if they aren't being that hard doing it. Biotechnology tends to start more from having a scientific hypothesis and designing around that. And there wasn't really a biotechnology industry until Genentech got started up north back in the 70s. And that's about applying what we can learn out of biology and making products out of it rather than just going for pure chemistry, like how the pharmaceutical companies started.
So how have you taken biology and how have you been able to simulate that with modern technology?
So I think Emerald Therapeutics and Cambrian Genomics are really good examples of that. Those are both companies that are just a bit north. And they're designing new tools to allow for biology to happen much, much faster in terms of its research. So Emerald has now what's called a cloud laboratory, where up in South San Francisco, they've built out a giant warehouse full of robots that move tiny bits of fluid around, because PhD students are actually like highly paid to move tiny bits of fluid around, and that's most of molecular biology research, is moving expensive clear fluid from one tube to another and mixing it together. But they have robots that do this now, that you can make amino acids, you can culture your cells, you can image your cells, you can even print tissues to a degree. Emerald doesn't have printing tissue capabilities, but they can do it all from your computer. There's a lot of startups going after that space where they're applying robotics to allow people to get into biotechnology research much, much easier. I'm new to the area, so I'm not entirely sure of it yet, but I think there's even a biohacker space about 20 minutes away that has open tools for everyone to get a hold of and try to do interesting science with. And last I heard, they're actually working on an open source tissue printer, trying to actually print kidneys or even bladders or bone.
For making like kidneys and bladders and bones, what are the actual like printed organs made out of?
So polylactic acid is the most commonly used material, as it's biocompatible and will degrade over time. But there are a lot of other biopolymers that are used in printing that have to be non-toxic, which grade well. And it turns out that the rheology of that material matters a lot. So rheology is how does it flow and how does it stretch and balance. And it turns out that if you can have variability in the structure, you can get the cells that you're then putting on that to differentiate much, much better. So traditionally, in printing tissues, they've been limited so far to bones and bladder because of how simple they are. With a bone, you can either decellularize an animal tissue so that you have just a collagen matrix to grow cells on, or you can print something new, but you don't have to have a lot of variation in that material for a bone to grow well. And that's somewhere actually where South Korea is about 3 years ahead of the US in terms of having products on the market and doing those kinds of things. There's about a dozen companies there that actually have bone products where if you have a major fracture or have to do an internal amputation due to something, they can print in kind of a reslotted bone and recolonize it with your tissue so it grows back to normal. And the bladder is basically just a bag of skin, for lack of a better term. So it's fairly easy for it to grow, but the kidney is really complex. If you look at a kidney on a cross-section, it looks like marbles connected by spaghetti. And it's a big mess on the inside in terms of what it does. And it's basically a filter. And printing a kidney is something no one's really done yet. They've been able to print what they call kidney organoids, really tiny versions of kidneys. And they've done that by printing a couple of different materials with different elastic properties, and then seeding that with different cell types. Because if you're going to 3D print a tissue, you have to have the substrates, and you also have to have the cells that go onto that substrate. So there's a company called Biotech that actually makes what's called the BioPlatter. It's the most advanced cell printer out there right now, and it has like 4 different print heads that you can lay down 2 different types of scaffold and then 2 different like cell groups if you want to print like 2 different cell types that then differentiate and create an organ on the other side.
So it's essentially like an artificial organ?
Yeah. And you then have to culture it in liquid and feed it nutrients and guide its differentiation before it's ready for implant. And in the cases where they've done bone implants in patients so far, they primarily, they get it going to a certain spot in the dish, and then they actually implant it like under the skin in the thigh or in the abdomen for the body to become familiar with it, make sure it's not going to be rejected, and to finish colonizing and revascularizing that tissue before then removing it and planting it elsewhere. I think the biggest one they've done so far is a complete lower jaw.
So it's just like someone grew a jaw somewhere else in the body, then they put it. It's pretty cool.
Yeah. They had the two parts of the jaw growing in that guy's abdomen for a while, and he had had a really, really bad accident. I think it was a burn problem, where tissue just went, and he was left without a jaw, which is not a great place for anyone to be.
And how long would it take for a jaw to grow? Because it's fairly big, right?
Mm-hmm. If I recall correctly, reading that report, he had that implanted for 4 months before it was ready for surgical reconstruction.
Wait, so can you clarify that process a little more? Like, does the actual jaw grow in a different part of the body, and like, how do they make sure it grows in that shape?
So to clarify that, yeah. So what they did in that is they took a jaw shape that they printed out on a scaffold,
Okay.
and then they took that patient's cells, and they modified those cells, grew them up, and then kind of like seeded them onto that substrate.
Okay.
It's kind of like putting grapevine on a trellis when you're making wine to guide its
Okay.
growth. And they did that to a certain point of development in a dish, but it can't get past that. So then they had to implant it into him for further
Okay.
growth and development before actually putting it where it needs to go in the end.
So the substrate is not inside, so the substrate is not inside, you said growing where did you say it grows?
So they grow it initially outside of the body in a dish on
Yeah.
artificial substrates, and then for a final processing, implant it into the body, for the body to become familiar with the immune system, doesn't reject it, and to finish vascularizing it, because getting angiogenesis, where you actually have a blood supply grow into a new organ, is one of the toughest problems in that field.
Do they have to use stem cells for all of these transplants and organs, or?
To a large degree, yes. So stem cells is an interesting concept in general. So there's a concept in stem cell research called stemness, where a cell is on a continuum between being differentiated and having a set function, and being more plastic and being able to be modified. And it was Shinya Yamanaka back in 2006 in Japan who discovered that he could reprogram adult cells into stem cells with 4 factors. So we have a couple different types of nucleic acids rather around within us. So DNA stays within our cell's nuclei for storage of information, but then for that to go out into the cytoplasm and become functional proteins, there's an intermediate called RNA. And what Yamanaka found was that if you introduce 4 RNA transcripts in greater than normal levels to any adult cell, you can get that cell to forget what type of cell it is and go back to being a stem cell. And from that, you can introduce other factors and redifferentiate it into something else. At Stanford recently, Joseph Wu, who's a cardiologist who does research, who found who's solved a problem in this field. The cardiologists have this issue where they don't know if a given cardiac drug is going to introduce additional heart arrhythmia in the patient they're trying to stabilize the arrhythmia in. So they kind of cross their fingers and hope for the best for the new patient. So what they did is they developed this interesting new assay where they take that patient's white blood cells and they de-differentiate them, so they make them more into stem cells. And then they introduce factors to those stem cells to make them behave like heart cells. And heart cells in culture, they have electrical things going on their surfaces, and they'll actually clump together and start beating. Like seeing that under a microscope is actually really weird. On like day 7 of the cardiomyocyte culture, they start beating no matter what you're doing. So they're reprogramming white blood cells into these beating cells, and then they have a specialized dish that has electrodes on the bottom, and they measure how fast those cells are beating. And then they introduce combinations of heart drugs to that individual patient's cells to see which of those things are going to introduce arrhythmia for that patient.
If you have to if people are starting to change adult cells into stem cells, does that mean there's a lack of stem cells?
So that goes back to the debate between embryonic stem cells and induced stem cells. So embryonic stem cells are considered the gold standard in the field, but there's ethical issues around obtaining them because you have to destroy a human embryo in the process of extracting those cells. And I think it was back in 2002 that George Bush put a moratorium in the US on any new embryonic-derived cell lines. And that really limited researchers on what they could do for a long time. However, induced pluripotent stem cells from adult cells kind of gave the field a jump start, because before they'd been limited by the sources of embryonic stem cells, but now they can make them from whatever. And with that, they can now kind of go off and do whatever research they need to. So there's not really a shortage of it. It's kind of the question is more, are these cells really behaving the same as the cells in our body or not? There's some differences in an induced stem cell versus an embryonic stem cell that may wind up being important down the line, but an induced stem cell is much easier to obtain and work with than an embryonic stem cell. So,
So returning to the earlier discussion on the growing the organs inside of your body after a point, how similar is that to growing organs inside of other animals like pigs or?
So it's a really similar process to using other animals as the host, and there's been a lot of work in immunocompatibility with pigs, trying to make it so that you can grow it up in a pig and then put it into a human and not have an issue, especially with hearts. But the issue tends to be immune rejection. Because pigs are so different on a molecular level, our immune system innately rejects that. But there has been a lot of work in pigs in particular of taking a pig heart and stripping all the pig cells away from it so it only has a collagen matrix left in the shape of a heart, and then recolonizing that matrix with human cells to replant it, because there's a huge shortage of available heart transplants.
Yeah, I've heard something about like some automation lab like space for like bioengineering. So it's like basically a space totally automation, and like the engineering or the bioengineering just program it, and all the like a robot inside of it can just do the things exact to any other engineering. So I just wondering why this kind of, is this kind of technology just start, or is it because still there's something humans can do but like the robot cannot do? So this kind of technology is still not expressed to all the world.
So it's beginning to get better and better. The Emerald Lab Laboratory, if you want to do genetic engineering of bacteria or of mammalian cell lines to make biological products, you can do all of that from a computer. And Emerald Therapeutics has actually built up a markup language similar to HTML for specifying your experiments, what order they need to be done, and how they need to be treated, what needs to be measured, and what goes into it. So you can do a lot more with that. There's several other companies working in that space doing basic bioengineering for like recombinant DNA in either bacteria or mammalian cells. But there's always going to be limitations to like how much has that laboratory built out. They built out what's called flow cytometry, which allows you to measure the surface of cells and what type of cell is this, what type of cell is that, how abundant are those relative cell types. But they don't have artificial peptide engineering. So if I wanted to go with Emerald Therapeutics for an experiment where I needed some completely synthetic peptide, I would have to source that from somewhere else, ship it to Emerald, and then get it done. But it's set to get much, much better very quickly.
So that means like it's because some technology limitations, so it still keep on working, but you cannot replace a real person right now, right?
You can, to a large degree. So with Emerald Therapeutics, you can, with your computer now, set up an experiment in about 30 minutes that would have taken me at the bench probably 12, 16 hours to carry out completely.
Right, definitely the calculation, but what about like the research? I mean, maybe for doing the test or doing things. Is that possible, like a totally automation doing that?
It will be. It's
okay.
not quite there yet.
Oh, okay.
So right now it's a matter of instrumentation. So what Emerald is doing is they're taking off-the-shelf instruments that are made by other manufacturers, integrating them in line with one another. And they actually still have a huge problem there of getting one sample from one machine to another machine, and they've hired technicians, humans, to do it. But they're getting much better at it. Or the Cambrian Genomics took the entire supply chain of DNA synthesis for making artificial DNA and completely automated it to make it much cheaper. So it's beginning to happen piece by piece, and it's kind of waiting for someone to come together and integrate all of these things together. But at the same time, instrumentation is changing quite a bit. Whereas before, like doing immune cell surface readings, I would have had to have taken an entire spleen from a mouse, which is about 20 million cells for me to measure. There's now companies working on at Berkeley that you can take 2 or 3 cells in the well and watch them over time, and actually have real-time measurements of how that cell is changing. And that kind of technology, as it gets more and more integrated with other things down the line, will allow more and more automation of that process. There's another side to that, though. It's called the Contract Research Organization ecosystem. So Contract Research Organization, or CRO, is a type of company in biotech that specializes in doing a kind of assay for drug development. And they may specialize in doing like hepatocyte toxicity tests, where they take liver cells from multiple different species and test, is your drug going to screw with liver enzymes and toxicity? Like that's one specialty. But now that since about 2006, there's been a huge diversity of CROs being started and specializing more and more. So it's now, for the first time, possible to start a biotech company and not have a laboratory of your own. You can do it all from your computer and a desk, and through email, set up experiments with CROs, get your compound, ship it out, test it in various places, and do all your development virtually. You don't have to hire any scientists, you don't have to buy any equipment.
Take a couple more questions, guys. We've about 5 more minutes.
I'm not sure if you covered this earlier on, but what was your background? What led you to join this kind of field?
I touched on briefly. So I studied cell microbiology at University of Michigan and got really interested in immunology. Took classes at the med school and was honestly a terrible student because I skipped so many classes to be in laboratory. I spent like 60 hours a week in the laboratory and maybe 10 hours a week in class. So maybe not the best balance, but I learned a lot that way. Because the question I have for you then is, knowing what you know of your own biology right now, like, is there something that you would change about it? Is there something that you would rather switch out, improve? Based on like, just your living day to day and what you have in your own bodies, can you see room for improvement?
Appendix, we don't need that.
The appendix actually has a pretty critical role.
Okay.
So we have our gut microbiota, which there's a whole ecosystem going on inside of our guts. So after an infection, the appendix repopulates us with healthy bacteria.
So then when it inflames and gets removed, what do we lose?
So we lose that reservoir of healthy bacteria.
So what risk does that pose to our...
It means that you're more susceptible to changes in your gut microbiota, which have been linked to a variety of other conditions. There's actually been some research showing that people who have an overabundance of a type of bacteria called firmicutes are more susceptible to certain kinds of allergens and developing long-term allergies against those. Whereas if they have more of an aspergillus flu, they're more resistant to those allergies.
Actually, I have another question too. Can we relate this to like neuro stuff? So like people with memory loss, can we do anything about that with the sort of fields you've been talking about?
I mean, potentially we can down the line, but memory is still pretty poorly understood.
Yeah.
Like the actual mechanism by which we form memories and where they're stored is still not well understood at all. But I mean, that's the thing that I really like about microbiology in general, is that if you look hard enough at any aspect of it, you're going to find big things that we don't just have no idea on yet. And there's a lot more to do and a lot to think about within that.
Any parting words of advice for life, for college, for career?
I'd say in terms of career, just keep your big picture in mind. For me, it's always been about making sure I have the ability to kind of form research projects on my